Welcome back, Biomonsters. Today we're going to be talking about meiosis. All right, let's do some quick review before we start talking about this new concept. The last time that we met, we talked about the cell cycle in general. Specifically, we also talked about the division of the nucleus and the cytoplasm to make two identical new cells in the process of mitosis as well as cytokinesis. Today, we're going to use that information that we learned in our last lecture to build on it to figure out why some things happen differently and how we need to make modifications in terms of cellular division. So let's dive into our review. The first thing that we want to make sure that you know is that mitosis produces two cells with the identical number of chromosomes as the parent cell. So we're making essentially twin cells in the process of mitosis. Another thing that's important to remember about mitosis is that this only happens in somatic cells, also known as body cells. So for instance, skin, stomach, heart cells, kidney cells, cells that line the mouth, all of these cells are somatic cells because they're body cells and they have nothing to do with your sex. And these cells all make extra copies of themselves in the process of mitosis so that they make identical cells every single time. The next thing that we want to review is how many chromosomes are actually found in each one of your somatic cells. Remember, every species has a magic number. Our magic number happens to be 46 chromosomes. And we can even take it further and say we have 23 pairs, which means the same thing. So here's what we mean when we're talking about mitosis. If I have a starting cell that has 46 chromosomes in it, and I go through the four phases, which are prophase, metaphase, anaphase, and then the last phase, which is telophase, and the easy way to remember that is to think about PMAT, because it has to go in order. If I do this correctly, if I started with 46, I should end up with two new cells that still have that same number of chromosomes because we're making identical cells or we're making twin cells. It's really important. So when these two numbers match, we know that we've done mitosis. But there is a problem. All living things have to be able to reproduce. We know that reproduction is one of the 5 characteristics of life that we talked about at the very beginning of the year. But we don't reproduce asexually, and we know the last time we talked about mitosis that mitosis is used only for asexual reproduction when we want to make clones. Well, humans, we don't want to make clones. In fact, there's no other person exactly like me on the planet, and the same could be said for you unless you have an identical twin brother or sister. So we don't reproduce asexually. Instead, we reproduce sexually. So that means in sexual reproduction, that it provides cells to make offspring. So we have to have a special process to make our specialized cells that are used in sexual reproduction. So it's going to be a little more sophisticated than the process of asexual reproduction that we saw in the amoeba earlier in our last presentation. So here's an example. In sexual reproduction, we need to take one cell from mom and also another cell from dad. So let's say that this cell is coming from mom and this cell is coming from dad. If mom and dad both had 46 chromosomes in their cells that they use for sexual reproduction, there would be a problem because inevitably we'd have to add those two numbers together and their baby would be starting off with 92 chromosomes in each cell. Well, that is a bad situation. In fact, it's lethal. Baby would not be able to survive with 92. We need to find a way to create a new generation or the offspring using two cells, one from mom and one from dad, that still maintains the magic number for our species. The answer to this specific problem is to ensure that not all of the cells in the body have 46 chromosomes. We just want to make sure that the cells that are used for reproduction, specifically sexual reproduction, have a different number. So that means sex cells, or these guys are also called gametes, only have half the number of chromosomes that are found in your somatic or your body cells. So if they only have half, instead of having 46, instead, gametes are only going to contain 23. So our gametes or our sex cells would only contain 23 chromosomes, while our somatic cells or body cells contain 46. So we're going to use two different types of cells for these two different processes. So here's your first stop and jot. Go ahead and pause the video. Use your notes to help you answer the questions on your day sheet, and when you're done, raise your hand so that your teacher can check your answer for correctness. Now that we've talked about gametes, and we know that these are specialized cells found in the body that only contain half the number of chromosomes that are found in your somatic or your body cells, we need to talk about what these cells are actually called. So humans, believe it or not, actually produce two different types of gametes depending on whether they're male or female. Males produce a specialized gamete called sperm cells in their testes that are used for sexual reproduction. Women, on the other hand, produce something called an ovum or an egg cell that are manufactured or made in the ovary. So those are our two specialized gametes that only have 23 chromosomes. So now let's see if our math adds up. If we have a cell from mom, so let's say that this is from mom, and if it's from mom and it only has 23, we know that it's a gamete. So this specific gamete is going to be an ovum or an egg cell. This one is coming from dad. It's still a gamete because we know that it only has half the chromosomes that we need. So this one is going to be a sperm cell. If we add these two together in a process of fertilization, it's when we bring egg and sperm together, we should get a baby that has the magic number. And our magic number here again is 46. This is our magic number for our specific species. And the only reason we were able to maintain this number is because dad gave 23 chromosomes in a sperm cell and mom gave 23 in her ovum or her egg cells. Now, the next question you're probably asking yourself was, well, how do we get a cell to go from 46 to 23? And that's a great question, and that's what we're going to be focusing on today. So when gametes are formed, their chromosome number has to be divided by half, or we can say reduced. This process occurs during a special set of cell divisions called meiosis. So we can call it reduction, division, so reducing the number of chromosomes happens in a special process called meiosis, similar to the word mitosis. So meiosis produces gametes with half the chromosome number of the original cell. So for humans, we would start with the chromosome number of 46, and we would divide it and bring it down to 23. So that's going to be our goal for meiosis. We just need to figure out what those steps actually are. Now that we've talked about the process of meiosis, we need to actually discuss some new vocabulary terms so we can understand the subphases that allow a cell to go from 46 chromosomes down to 23. So a cell that only contains half the total number of chromosomes is referred to as haploid, which makes sense because haploid almost sounds like the word half. Haploid cells, also known as gametes or sex cells, are represented always by the letter N. So it's going to be our placeholder. A cell that contains the total number or the magic number of chromosomes for a species is referred to as diploid. And our diploid cells, or our body cells, also known as our somatic cells, are represented by the number 2 and the letter N, which makes sense. So 2 times the haploid gives us the diploid number. Here's another way of thinking about it. So if our chromosomes are 46 in a cell, the vocabulary term that we would use for that is the term diploid, and the abbreviation would be 2N. If instead we had a cell that only had 23 or half the number of chromosomes, we would call that cell haploid, and we would abbreviate it by by the letter N. So now let's do some practice problems with our vocabulary. In a fruit fly, or if a fruit fly has 8 chromosomes, so this is important, in its intestines, what is its diploid number? So we're looking for the diploid number, and we should already know that diploid equals our 2n number. Well, the first thing that I have to ask myself is whether or not the number they gave me is diploid or haploid. And the way that I figure that out is I need to figure out, is this 8 found in a type of cell that is a body cell, or is it found in a sperm or an egg? Well, if I read here, I notice that it tells me it's found in the intestines. Intestines are body or somatic cells. And body or somatic cells are always diploid or 2n. So I don't actually have to do any math for this word problem. I should already know that my 2n number is 8. The next question is, how many chromosomes would be present in the fruit fly's ovum? Well, in order to figure that out, I have to ask myself, ovum, is that a body or somatic cell, or is it a gamete or a sex cell? And I know that ovums are the female versions of sex cells, also called gametes. And I know that my gametes are always N, or they're haploid. So in order to determine my haploid number, all I have to do is take my 8 and divide it by 2. So my haploid number in this case for my fruit fly would actually just be 4. So here's your first stop and jot. Go ahead and pause the video. Answer the questions on your day sheet. When you think you have the right answer, call your teacher over to check for correctness. All right, now let's talk specifically about meiosis. Meiosis is a reduction All right, let's talk some specifics about meiosis. Meiosis is a reduction in chromosomes so that gametes become haploid. Remember, haploid means half. There is a two-way process that has to happen in order to bring our number from 46 down to 23. So meiosis actually has 2 divisions, which makes it different than mitosis, which only has 1. So the 2 divisions are meiosis 1 and meiosis 2. In meiosis 1, homologous chromosomes are separated, so they're 2 chromosomes, remember, that are roughly the same size, shape, and control the same function. And in meiosis 2, we're separating chromatids in each chromosome in order to bring our final number down to half of what we started with. So here's what it looks like. The meiotic divisions are very similar to the meiosis divisions that happen, or the meiotic divisions 1 and 2 are very similar to the terms that you saw in mitosis, and in fact, a lot of the similar activities are going to be happening as well. So in meiotic division number 1, we have PMAT1, and in meiotic division 2, we have PMAT2. So think about the things that happen in prophase, metaphase, anaphase, and telophase. Similar events are also going to be happening in the 2 divisions of meiosis. So in prophase, the nuclear envelope is going to be disappearing. Chromosomes, or DNA, is going to go from a loose structure to a tightly wound package structure in a chromosome. In metaphase, things are still lining up in the middle. In anaphase, we're still splitting things apart. And in telophase, we're just undoing what happened in prophase. All right, so here's an example of what's happening in meiosis 1. If we look at that very first picture at the top, you'll notice that our nuclear envelope is starting to disappear. Our chromosomes have started to form unique, our DNA has started to become chromosomes, so they're uniquely organized and easy to divide. But they're also going to line up in pairs. You'll see that these 2 are roughly the same size and shape, so this homologous pair will line up. These 2 are roughly the same size and shape, so they'll line up as well. So that's prophase 1. Now in metaphase 1, you'll notice that our homologous chromosome pairs are lining up together, but they're still found in the middle of the cell. So that's going to be metaphase 1. In anaphase, just like in anaphase and mitosis, we're going to be breaking things apart and moving them to opposite ends of the cell. The same thing is true in anaphase 1. The only difference is we're not breaking apart chromosomes, we're breaking apart homologous chromosome pairs. Lastly, you'll see that we have telophase 1, and in telophase 1, we're basically going to recreate our nuclear envelope, and we're going to be separating out our 2 piles of genetic material, but they're going to be identical or roughly identical to one another. And then lastly, we're going to have, the last thing that's going to happen is cytokinesis, where we're going to actually pinch our cell into two new cells. Now, what's unique about this individual process is that in meiosis, we're going to have a unique process called crossing over happen. Now, crossing over is really important. It occurs when portions of the chromatids on our homologous chromosomes are swapped with a chromatid portion of another homologous chromosome. So here's what it looks like. So here are two homologous chromosomes. They're going to swap out pieces. And when they swap out pieces, they're no longer genetically identical to one another. Now, this process is really important. If we're comparing this chromosome now to this chromosome, they're no longer identical to one another. We swapped out a portion here, and we also swapped out a portion here. That means that when we're actually looking at the first division of meiosis, we're not creating genetically identical cells. Instead, we're intentionally adding genetic variation. So once the chromatids are genetically different, or crossing over has happened, when the chromosomes separate, they're going to form two new haploid gametes that will be genetically different from one another. And this is important. This explains why if you have a brother or a sister, they're not exactly like you. You might have some things in common with them because you have the same mom and the same dad, but when mom and dad produce their egg and their sperm cells, every single egg and sperm cell that they create is going to be genetically unique because crossing over took place in the first meiotic division that allowed them to actually develop. So you are genetically unique, and you will never exist again on the planet because mom and dad will never be able to create the same egg and sperm that was used to make you on your birthday. All right, let's talk about meiosis 2. Now, meiosis 2, we're still doing the same thing. We're still doing PMAP, but we're doing the second phase of PMAP. So if you look here, you'll notice that we're doing this twice because our two cells that we got from our first division need to divide in the second division. So the first thing that's going to happen here is we're going to take our cells and we're going to start to break down the nuclear envelope and ensure that our DNA is tightly packaged into our chromosomes. And you'll notice that our chromosomes are no longer genetically identical, so these two cells are unique because crossing over took place in meiosis 1. After prophase 2, metaphase 2 is going to happen, and we know that this is metaphase because, look, all of our chromosomes are lined up along the equator or the middle of the cell. Then we're going to go into anaphase 2 where we actually separate the chromatids, the sister chromatids, from one another. And once we separate them, we're going to reform our nuclear envelope, and notice that we're now creating 4 nuclei. And then the last thing that's going to happen is cytokinesis, and when cytokinesis happens, we're going to end up with 4 new cells. And these 4 cells are all haploid and genetically unique. All right, so let's talk about what's actually happening here. Our nuclear envelope is going to start to disappear. Our loosey-goosey DNA is going to start to coil into our homologous chromosome structures, and they're going to line up with one another. We know that they're going to cross over. That's an important feature of meiosis that creates genetic variation. Once we have crossing over take place, metaphase 1 is going to happen where they're going to line up at the equator. And now anaphase 1, they're going to be pulled apart. And in telophase 1, we're going to reform our nuclear envelope and keep them separated. And then finally, cytokinesis is going to happen. So we now have 2 new cells that are haploid, and they are genetically unique. Now we're going to move into prophase 1, or prophase 2, metaphase 2, and anaphase 2. Finally, here we are in telophase 2. We've had cytokinesis happen for a second time, and we now have 4 haploid cells that are all genetically unique because of crossing over that took place in the very first division. Here's your next stop and jot. Go ahead and pause the video, see if you can answer the questions on your day sheet, and when you think you have the right answer, call your instructor over to check for correctness. All right, now that we've officially done talking about meiosis, we want you to be able to compare meiosis and mitosis to one another. So here's a quick little video recapping mitosis and meiosis.
Prior to meiosis or mitosis, DNA replication occurs. In both meiosis and mitosis, the chromosomes condense and become visible under the microscope. In meiosis, homologous chromosomes pair up or synapse, and crossing over takes place. Homologous chromosomes then align themselves along the metaphase plate, and each homolog is drawn to the opposite end of the cell. In meiosis, a second division occurs, resulting in production of 4 haploid cells. A single-cell division occurs in mitotic cells, resulting in 2 diploid cells.
Some other things to remember about this process as we're looking here, you should also note, so we have meiosis makes 4 cells. All of these cells are haploid, and because they're haploid, we know that all of these guys are actually going to be sex cells or gametes. So meiosis makes 4 cells that are haploid, they're sex cells or gametes, and we also know that these are all genetically unique, and we know that they're genetically unique because crossing over has taken place in meiosis. Now on the flip side, if we're talking about mitosis, mitosis only makes 2 cells, but the 2 cells that it makes are all going to be body or somatic cells, and these somatic cells are all going to be genetically identical. Another thing to keep in mind is that mitosis only happens in when we need to repair things, when we need to grow, or when we need to do asexual reproduction to make clones. Meiosis only happens when we need to do sexual reproduction. All right, here's your next stop and jot. See if you can answer the questions on your day sheet, and when you think you have the right answer, call your teacher over so that he or she can check for correctness. All right, now that we've talked about mitosis and meiosis, and we've talked about the best case scenario, what you do need to know is that sometimes these processes go wrong. So what we need to do is talk about what happens when there are actually problems with chromosome numbers. And if you look on the slide in front of you, you'll see that there is a picture of chromosomes laid out based on size and pairing. So you should remember that this guy is called a karyotype. Now, remember, karyotypes only help us do 2 things. They tell us whether or not the individual has the magic number of chromosomes, which should be 46. It also tells us whether or not the individual is male or female. That's the only thing a karyotype can tell us. It can't tell us how old the organism is, can't tell us what color eyes, how tall they are, so it only tells us if they're male or female, so their sex, and whether or not they have the appropriate number of chromosomes. There is, unfortunately, a number of genetic conditions that can be identified by looking at a karyotype. And the first genetic condition that we're going to focus on, the main one that you need to know, is something called trisomy 21. So trisomy 21 is a genetic disorder caused by an extra 21st chromosome. So someone with trisomy 21 actually has a total of 47 chromosomes. And in this case, believe it or not, it's never a good thing to have more, and it's also not a good thing to have less. Trisomy 21 is more often commonly referred to as Down syndrome. And you probably have met someone or known someone who has Down syndrome because Down syndrome causes a characteristic number of symptoms and phenotypes or physical expressions of that particular genetic disorder that are easy to point out. So if you look at this picture of this little baby here, you'll notice that they have similar characteristics in terms of the fat deposits on their face, in terms of the way that their eyes are shaped and how close set they are to one another. So here's another picture of a karyotype, and what I want you to do is study this karyotype and see whether you can tell me, is it a male or a female, and then also tell me whether or not you think this is a genetically normal individual. Well, if you studied it closely, you should have noticed that if you're looking through, this looks good, we keep going, this looks good, but finally we get here. And when we get here, you'll notice that this is the 21st pair, so I could count them all up, but this pair has 3 chromosomes instead of only having 2. So we know that this individual has trisomy 21 or Down syndrome, and trisomy, tri, simply means 3. It means that you have an extra chromosome on your pair. Finally, we can look at the sex chromosomes. So these are my sex chromosomes that determine the gender. My sex chromosomes on this karyotype show me that I have an X and I have a Y, and you should remember that Y means guy. So this happens to be a karyotype of an individual with Down syndrome who happens to be male. The last thing that we're going to talk about are changes in chromosome structure. So changes in an organism's chromosome structure are referred to as chromosome or chromosomal mutations. And there are 4 types of chromosomal mutations that we're going to talk about today, and your job is to be able to identify them via picture and also by word. So the 4 types are called inversion, translocation, deletion, and insertion. So let's go ahead and talk about each one of these. So our first one is inversion. In chromosomal inversion, what we're going to do is we're going to take our chromosome. So I'm going to draw one, and you can draw one in your notes as well. So our chromosome should look like this. And I'm going to draw a smiley face here, and you'll see why in a second. In inversion, a piece of a chromatid breaks off and flips, or we can say inverts itself. So let me show you what that looks like. If this is the before, so this is what we should have, so this is what we should see in a normal chromosome, here's what happens after the mutation. So instead of the chromosome looking exactly like what you see in the original picture, we're going to take a piece of our chromatid that broke off, so here it is, and the smiley face is now upside down. So this piece broke off and flipped upside down. So that's what a chromosomal mutation looks like if it's inversion. Translocation is a little different. In a translocation event, one piece of a chromatid from one chromosome breaks off and reattaches to another chromosome. So here's what that would look like. If this is my chromosome, this piece right here would break off. So it's no longer there. And when it breaks off, it's actually going to travel and it's going to reattach itself onto another chromosome. So when it reattaches itself onto another chromosome, it'll look something like this. So now this chromosome actually has extra genetic information, while this chromosome is missing genetic information that should be there. So that's our translocation event. Our next chromosomal mutation is something called a deletion mutation, and its name pretty much tells you what it does. So a piece of a chromatid breaks off, and that's it. It doesn't go anywhere else. It doesn't reattach itself. So what does that look like? If this is my chromosome, and you guys should be drawing these pictures on your papers as well. This is my chromosome. A piece, let's say this piece right here, breaks off so that we end up with a chromosome that now looks like this. So the genetic information that should be here is now disappeared or it's been deleted. And our last chromosomal mutation is something called insertion. So an insertion event is simply breaking off a piece of one chromatid and simply reattaching it to the same chromosome at a different location. So here's what that's actually going to look like. So here's my chromosome, my original chromosome, and my original chromosome will have some sort of event that will happen that will break off this little piece of my chromatid. When that little piece of my chromatid breaks off, we're now going to see a new chromosome, and my new chromosome looks like this with that piece missing. But what I'm going to do is reattach it to the same chromosome, and it's just going to go somewhere else. So it might attach right here onto the other chromatid. So that's my insertion event. All right, here's your last stop and jot of your meiosis notes. Please answer the questions on your Cornell notes. And when you think you have the right answer, call your teacher over to check for correctness. Be sure to study your notes in preparation for your SOL as well as your upcoming exam. Thanks so much, guys.